Tom is on the call, I think temporarily. He is Tom Alabama's in right now, so he's with his family on spring break. But he's going to at least join us for a little bit. So welcome. Let's see. So I can talk a little bit about. Paul said most people have what's happening in the future for PLATO, for the PLATO rundown. Like next week is going to be Reba Loiken, probably fingers pronouncing her name, the director of Allen Centennial Gardens. will be here talking about the future of Allen Centennial Gardens. The week after will be Dave Nelson. But tonight, for Wednesday night at the lab, we are going to be talking a little bit about coronaviruses. So we have Robert Kirchdorfer, who's going to talk about the molecular biology of coronaviruses, if you'd like to join us tonight. Yeah. And then next week, not aware, is our large open science open house that is happening on campus for April 8th, 9th, and 10th. And there will be a special version, Friday night version of Wednesday night in the lab on April 8th, where they're going to be talking about the discovery, recovery, and curation of the 1,200-year-old dugout canoe from Lake Mendota. So you can join us for that special Wednesday night at the lab as well. But I think, Paul, you have anything to say unless we're ready to go. On. All right, so this afternoon we have, I'm going to let Maria pronounce her last name. And she actually is going to talk a little bit about her last name as well, I believe, in the presentation. So I'm going to pass it on. Welcome.
Thank you. So much for the invitation. Today I brought you guys a talk with a very eye-catching name of microfluidics and cancer modeling. So first of all,
Started.
All right.
Okay.
That's coming in now. Okay, so I wanted to introduce myself a little bit more first, tell you like where I'm coming from. My name is Maria Virumbrales Munoz. I'm originally from Spain. I have two last names, no middle name. So I typically get a lot of confusion, so I just wanted to put it out there. I grew up in the lovely town of Logroño, which is around there. And I did part of my undergrad, and then I moved to somewhere called Zaragoza. And I brought a couple of pictures for you guys to know. This is one of the most iconic streets of Logroño. And over there, that is El Pilar, the Cathedral of Zaragoza. So I did my Bachelor of Science in Biochemistry, and then I have a master's degree in molecular and cell biology, a master's degree in teaching. And then I went into my PhD in biomedical engineering straight away. That took me 4 years. I spent a couple months in the UK and the Netherlands in that time. And then in 2017, I moved here to Madison, and I've been working as a postdoctoral associate ever since in Dr. Beebe's lab. Let's quickly raise hands over here. I want to hear about you guys. Who over here has a background in the medical sciences or medicine, something like that? All right. I love when you guys keep me on my toes. So that's fantastic. Do we have people here with, do we have people over there with a background in the medical sciences commenting?
Let me see if we have anybody in the chat.
All right. What about in engineering? Do we have any trained engineers in the room?
Not here.
All right. Great, because I'm not an engineer, hopefully I won't say anything that doesn't make sense. Okay, so there's that. So today I'm going to be talking first about what is microfluidics, and I'm going to start with a brief history of what that's like. Please feel free to stop me throughout the presentation. I have been told that you guys like dense presentations full of content, but I wasn't sure who I was going to be dealing with today. So if you guys feel like that things are not making sense, please let me know. Then I'm going to talk about why bother making things small, which is the whole point of microfluidics. Then we're going to be moving on of why, like, how do these models fit into the general science panorama? How do we pick the model that makes more sense for our experiment? What are some device building techniques? Hence, although toys that I passed out a moment ago. And then we're gonna be talking about like how far has the field come? And finally, I'm gonna be talking about two projects that I have been working on and that are not published. They're both gonna be talking about cancer and the lymphatic system. So we're gonna be learning a little bit about that too. Okay, so first of all, we're gonna be talking about what is microfluidics. And microfluidics is, very broadly speaking, the field of liquids that are handled under the micro scale. So over here, we had a scale of volumes from like an Armstrong to like a ton full of liquid. So typically our experiments in a normal lab will happen in this range over here. So from 1 milliliter up, but microfluidics handles these kind of volumes that are a lot smaller. And the interesting thing about that is that at a macro scale, conventional fluidics work in a way that is very different from microfluidics. And that's just a little teaser for you guys. We'll talk about that a little bit more. Talking about the history of microfluidics, the historians of the field say that it was inspired by one of Richard Feynman's talks, which was called "There's Plenty of Room at the Bottom." And they also talk about, like, well, he talked in that talk about how, computers at that time were like a room full of stuff large to make them work, but how we could make them progressively smaller and smaller and end up by manipulating atoms. And well, the scientists right now are saying that that really inspired the field of nanotechnology, but I come from microfluidics, so I want to tell you that it also inspired microfluidics. And as I was sort of hinting at before, microfluidics comes from the field of microelectronics. The same techniques that are used to fabricate electronics that now go into your cell phone or your computer are the same that we use to fabricate our devices that I passed out a little bit ago. Now, the field doesn't really exist until the 1980s, when we started to be able to manipulate liquids at a very small scale using micropumps and microsensors. And then from there, from the 1990s, as you can see over here in this timeline, it has really exploded. We started to call it microfluidics. We started putting cells and live organisms into these devices that were made with techniques that were used for microelectronics. So from there, we did a little rebranding of sorts, and we started calling ourselves the organ on the chip community. And that is because we realized that by using these very small structures, we could start imitating the functions of an organ using a very, very small system. That was in the 2020s. And I have over here a great graph talking to you about the number of papers that were published in microfluidics and cancer over the years. We started in 2005, and then you guys can see that it rises, it shoots up to like 500 a year. And then this graph stops at 2018. And I couldn't find the piece that's missing because it's all older. But over here, I did find a graph that's talking to us about how much money was being put into the field over the years. And that one starts in 2013, and you can see that it shoots up in 2024. So this is a field that is only going to get bigger. The NIH, the National Institutes of Health, has recently started putting money directly into these projects because they think they're out to change the world. Okay, so in the 2010s, very recently, we decided to rebrand again and called our devices microphysiological systems. And the only difference with before is that we're no longer just aiming to reproduce tissue or organ level functions. We're also aiming to reproduce disease and try to model disease, how it develops, and how we can treat it. So this is where we're at right now.
Where's the Wisconsin University on this scale of involvement?
That is a great question. So I initially had a slide over here that I just took out about who were the pioneers in the field. The pioneer, the grandfather of microfluidics was George Whitesides. And then there was a really strong first generation of engineers in microfluidics and all that jazz. And among those is David J. Beatty, who is my boss currently. So he has been one of the pioneers in the field with a very strong biological interest. Right now, we're one of the few labs in the world who is really putting most of their energy into this field. So there's that. I don't really have the numbers, but if you look at the literature, we're one of the top places where microfluidics and cancer modeling is being done as of now.
And what are you in engineering? Are you in what?
I trained as a biochemist / biologist of the cell and molecules, but during my PhD, I sort of converted into engineering. So now we call ourselves biomedical engineers, but typically you just train in one thing. Well, now the new kids are training in both, sort of the convergence of the both disciplines. But most of us have been doing research for a little bit in this field.
And where is your lab?
My lab is it's not my lab. I'm still a postdoc. Well, hopefully that'll change soon. But we're located at WIMR, Wisconsin Institutes of Medical Research, which is by the hospital. So we have direct contact with them, and that'll make sense as to why that's important during my talk. Do we have any more questions? Yes.
Yeah. Back in my day, we didn't have biomedical engineering. It was medical physics. What is it really the same thing, or are they?
Yes and no. I would say medical physics is probably a piece of that. When I hear medical physics, I think more about imaging large complex systems or studying the laws under which those operate, rather as engineering them from scratch.
Okay.
So a top-bottom versus a bottom-up, if you're from the engineering school of thought. Like we like to build rather than break down and study. Is that making sense?
Sure.
All right. Well, I'm an engaged crowd. Okay. All right, so let's talk about why bother making things small, because I have been talking about microfluidics as it just makes sense. Well, there's a few reasons why it makes sense to make things small. And the first one, very obvious one, is we use very small working volumes. And there are a few reasons why that's interesting. The first one is if we're trying to use stuff that is really expensive, as it happens in the biomedical field, and I don't think I need to tell you guys about that so much now in the COVID era, the less we need to use of those expensive reagents, the better. Now, something where it's also interesting is for really rare samples. The classical example of this are CTCs, or circulating tumor cells, which are those cells that get detached from a primary tumor and metastasize to find another spot and like start to grow and create metastasis. Those are really rare in blood, and there are very few of them every time we isolate them from blood of the patient. So not having to dilute those to study them, I believe it's a great advantage. And then this one is a bit more out there, but bear with me. There's a proximity effect. So when a chemical reaction happens, it happens because two molecules interact with one another at close range. So one would argue that if you are dealing with a very small volume, the things that are happening over there, all the molecules are at a closer distance. So the likelihood that those chemical reactions happen is a lot higher. The same happens when two cells want to talk to one another. If you have one cell over here, one cell over there, it's going to take longer for the message to come through, if ever. But if you got them really close by in a very small system, then the likelihood that they're going to talk to each other is going to be a lot higher. Hence, proximity issues and small working volumes. Oh, real quick, this is the first image that comes up of a microfluidic device. If you Google it, it's from a different lab to ours, but I thought I would just show some variety. And this is an example of application of one of those devices, where this lab, what they did is they cultured blood vessel cells on one side of this device, and then on the other, they put fibroblasts so that they could talk to one another. And you can see that after a few days, a branched structure of cells emerges out and tries to reach the site where the fibroblasts are at. So that's something that would be really hard to try and mimic in a traditional petri dish. That's something I'm going to talk about a little bit more later. All right. The next thing I'm going to talk about is the predictability of physics. I was already hinting at before a very stark difference between liquids at the macro scale, bigger volumes, at the micro scale, very small volumes. So I like to call it the magic of microfluidics versus physics. There is a point at which gravity matters less and capillary forces, diffusion, surface tension start to matter a lot more in these systems. And those are the forces that are actually driving how these liquids behave. And to me, that's really cool. So you guys might be familiar with this schematic. What this is talking about, that at very small volumes, in very specific circumstances, such as the human body, the liquids move in a very front-like pattern all at the same time, instead of in a turbulent and chaotic manner. And that's really important. That's sort of the same concept all over. And in biological systems, this is the way that liquids move. And hence, to me, it makes sense that we use micro systems to try and study how the human body works. All right, so I brought a couple of, well, one image, one video to illustrate these concepts a little further. The first one is our little bug friend over here. You can see that in that situation, gravity is not pulling the bug down. Capillary forces and surface tension are keeping it up, and hence our friend the bug is not sinking. And the other thing that I wanted to talk to you about, and let's see if I can actually share this. I don't know if they're going to be able to see that in Zoom. Can they see it through the camera?
Let's see. We can only see it. Let's see. Maybe like reshare maybe. I wonder if you do that, if they'll be able to see the video.
Okay, maybe we can put the link in the Zoom later.
Yes.
And then so they can.
Yes, in the chat. Yep. Sure.
All right. So over here we have a microfluidic system that overall is about 10 microliters of liquid, and it's colored red with food coloring. Okay. Oh wait, you guys can't see that either. Let me see if I can stop that for a second. I think I broke it. So let's that's such a shame. If I turn this, yeah, I think I broke it.
Interesting.
Okay, can you see something?
We're seeing it in the Zoom. We are seeing it in the Zoom.
Okay, so now people can see it in Zoom, but we can't see it over here.
Right.
That's not fun idea. Anyway,
But you're right, we could maybe flip it. I mean, we can maybe like, if we turn it, I wonder
Okay.
if like,
let's
and make it
try that.
full screen, then would people be able to see enough here? Okay.
All right, let's try and make this work. Okay, so we've got over here your 10 microliters of liquid, that red with food coloring. What's going to happen over here is you have two different colors of liquid. One of them is small, the other one is large. You'll have to believe me that this one is the small one and this one is the large one, because I don't think you guys can see it from there. Okay, so what happens over here is, again, capillary tension, capillary forces and surface tension are going to be driving the system. So instead of the big column pushing the small one down, it's actually going to go the other way. It's not gravity, it's going to be surface tension pushing the droplets. So if we now add a blue droplet of liquid on the side, the system was pushed from the small column to the big column. And I don't know if you can see it, but over here, we still have on the edges some red food coloring because the liquid is being pushed like so, but then in the edges, the red liquid still remains. So that is laminar flow. It's not turbulent and mixing. The mixing is not happening in the system. Now, something else that I think is interesting is that if we add a little bit more, you see that there's a piece, a small cloud in the middle of our original droplet that's blue. It's not all blue, it's not all purple. The center of it remains blue because there's no mixing. Over time, diffusion will equilibrate those two colors and everything will be blue/purple. But as of now, we have a piece that's red and a piece that's blue.
Why does that happen? That it goes
because there's no mixing. The liquids are moving in such a small manner.
From the small one to the big one.
From the small one to the big one.
Why?
And then they just don't mix because the capillary.
No, but why does the small one go to the big one?
The surface tension of the small droplet is actually very powerful. It's more powerful than the one in the large droplet. So that's driving the system. For that, they have to be connected to one another. But that force is the one that keeps pushing, opposite to what you would think in a normal, like bigger sort of systems. Yep.
What happens if you put the blue drop in the large part?
It moves a little bit. It's not as pretty.
It starts to swirl around a little bit.
It will continue to like bubble up.
Travel through to the other side.
A little bit. That will happen a little bit. Because at some point there will be no more room for the droplet to go. It's the same as if you continue to saturate the system from the small port. At some point, it's just going to have nowhere else to go. So it's not going to be able to balance the forces as well. Yeah, that is a great question. All right, let's see if we can put this all back. Back here. And then my only problem is going to be trying to turn this back.
The screen back on, yeah. Let me see, I can grab.
Ooh, looks like it's thinking.
Oh, taking it. Sorry. All right. How are they? Oh, it's back. Really? Okay, good.
Just walk you away, mid-work. Excellent.
How are we following things so far? Is this making sense? Mm-hmm. All right. Okay. So the next thing that I feel like is very interesting about microfluidics is our ability to control liquids in a very precise manner. By using different designs, different channels, different compositions of material, we're going to be able to generate small droplets of liquid. Like we can partition it very accurately. We can mix it. We can, I don't know, we can do tons of things. We can do droplets of two different colors, one after the other. So there's a lot of like tricks that we can generate by just controlling the system. We can also generate valves. We can generate micropumps. We can automate the system this way. And that's something that is really hard and very costly to do with a bigger system. And I think that's really cool. So all of this like high control and precision that we can do translates into biomedical applications because we can indeed, as you may have guessed, use these systems for biomedical application. And one of the most commercially profitable ones that have happened for these technologies is this little guy over here, which is actually about this size. And that is a quality control chip for RNA, which this day and age, everyone knows what RNA is. So imagine that you have a mixed bag of RNA that you don't really know what it contains. The best way to figure out how good that RNA is, if it's degraded or not, if you have different pieces, is to run it through one of these guys. You just input 1 microliter, 1 tiny droplet over there. And what that's going to do is something called an electrophoresis. It's going to be input into a giant machine, and it's going to apply to a current of electricity through, and it's going to separate all of the different pieces of RNA, which is intrinsically charged. And then it's going to give us with a very high precision how much we have, how contaminated it is, and what is the size of those pieces that we have. And these are expensive. So there's that. And then finally, I was talking about how we can really control the systems, how we can partition liquids. That is great, for example, for high throughput testing. Imagine that you have 1,000 new compounds that you want to test. A great way to do it is to use one of these situations in which you just say, hey, I have my bacteria 1 that I want to test 100 compounds onto. So I'm just going to divide my sample of bacteria into little bits with 1 droplet, and then in 1 other droplet for each of the drugs. I'm just going to add all of these drugs and see which ones are effective at killing that bacteria.
So Maria, I'm going to stop you for 1 second. For some reason, your PowerPoint is stuck on through the Zoom. It's stuck on there. I wonder why.
Oh my goodness.
And I'm sorry, I just realized it.
Yeah, too. I don't know.
You may have to reshare your screen, I think. I apologize.
I have 2 of them.
Ah, okay. That's better. Yep. And then I don't know if you want to do a full screen. It's showing it in a different mode. Perfect. Wait,
Yeah.
no.
Oh. Wait, so then here, right? Is that coming up?
Let's see. Not yet. No, now it's a black screen.
Does it change back or no?
I think it's probably fine if we can't fix it.
Okay, sorry.
No, that's okay. That's all right. Don't worry.
What is the picture in the upper left?
This guy over here?
Yeah.
So it's another version of one of the devices that we use. So over here, what we have are 3 different channels. You can see each of them in 1 different color. And then the way you access them is by these ports. So what happens, that's kind of an intricate design because it has layers in between. So you can access that one is sort of the upper layer of this channel. And then this one is the down layer of that channel. And they're both separated by a membrane. So you can put 1 thing over here, 1 thing over there, and the same one goes for all the other channels that are going on. And the total volume of this is, I don't know, maybe like 25 microliters, like a slightly larger droplet, like a raindrop.
Yeah, how do you machine these devices? Is it like with lasers or chemicals or
I
what?
have a couple slides for you.
Okay, okay.
All right. So let's talk about how these models fit into the general realm of the models that we know work for biomedical research. So you guys might know about your typical Petri dishes that are like different sizes, shapes that we use for typical in vitro, like into the lab is what it means, research. And then we've got a lot of like what we call model organisms. They can be mouse, they can be rats, they can be worms or flies that we do research with as well. Well, what I'm trying to argue today is that these models that I'm bringing are not meant to replace, they're meant to complement. There are some things that some of these models are better at. So the in vitro models are very simple. They're very cost effective. You can do a lot of them at the same time. You can use your human cells. But then if you go to your in vivo models, they're really complex organisms. You have a lot of things that you can look at, but then really figuring out why things are happening, what exactly is happening is a lot more complicated. And then on the other side of things, these are very cheap, whereas the model organisms are very costly. So the models that I'm bringing you here today, which in this slide are called organotypic models, which basically means they just look up a lot like an organ. They're in a very happy medium of that, but it's also sort of like a Goldilocks spot, because then there are additional things that these guys are better at than the other two models. For example, the integration of sensors, like you can couple one of these guys with a sensor, a light-based sensor, and you can start tracking them continuously. The handling of small samples that we talked about just now is great in these guys, and you cannot do the same in the vitro or in vivo models. That's not something that can happen. The volumes of things that you need are very small, all of that jazz that we spoke about a minute ago. And then trying to use these guys to generate complex structures that look a lot like the human body is a lot easier, I would argue, than in normal in vitro models. But they're also very controllable. So you can decide what are the pieces that go into your model, as opposed to in a mouse where everything is just crammed in there and you have to decide how you tease that out. And that's a really complex job. So again, we're not meant to replace anything else. We're meant to complement and serve as a bridge between those two very different worlds. So I brought here a case study. I specialize a little bit in vascular biology in cancer. So we're going to be talking a little bit about that. I chose to bring over here a selection of what some of those models look like if you look at them up close. Specifically for something called angiogenesis, which is the growth of new blood vessels from pre-existing ones. So what happens is you have a big blood vessel that starts growing, making branches, and then in the end it generates a new blood vessel. And it's a really important process in cancer. But what happens with this process is it's also really hard. It has a lot of phases. It has a lot of steps, a lot of things that are happening. So what I want to argue in this slide is that you're going to choose your model according to what it is that you want to study. So the first few models that we used to make using microfluidics look a lot like this. So this is media, and this over here, these tiny things are your cells. So you could just have a channel, and you would culture your cells over there, and everything else would be fluid that you could flow. You could also put them on top of a membrane. That way, for example, what you can study is how that monolayer lets substances pass through. And a change in that is really important in the angiogenic process, but it's only one of the events that happens. Something that we've come to do since then is sort of doing a combination of situations. So over here you would have, say, a block of jello, and then on the other side, you have cells cultured, right? So your cells now have access to a matrix that looks a little bit like the human body, where they can degrade, invade, and generate their branches. That's something that you couldn't do in this one. See where I'm going? So depending what you want to study specifically, you're going to choose what your model looks like. Right now, the models that we like to use a bit more are these guys, where you've got your cells, and everywhere else, there's jello, there's extracellular matrix, as we like to call it. And I'll come back to this point a little bit more later, but think about a block of jello where your cells are in the middle. So they can go everywhere. You can watch them from every side, and they're going to find something that they understand as relevant, a scaffolding that they sort of know everywhere. So you're going to be able to see them act and doing their stuff, doing their branches in a more physiologically relevant manner. And it goes on and on. Depending on what you want to study, you're going to be choosing one device, one model, or the other. Okay. So now we're going to talk about device building techniques, which is something that you guys have already requested. Okay. So as you may have imagined, these guys are really small, which means that they just can't be produced with your typical machining techniques. They have to be a little bit special. And the way that I kind of try to put them together is into three categories. One where you add material, one where you mold material, and your third one where you remove material. If you guys remember that little thin sheet that had some places that were transparent and the other ones were black, that is called a photomask. So what we do is we create a very thin layer of a polymer that is sensitive to UV light, and we illuminate only certain areas. And then we get rid of everything that wasn't illuminated, which we can do this first. Of sorts. So then that would be your first piece. And then you start building on top of that and on top of that. And you end up with a very complex design, sort of in a Lego style that you built from the bottom up. That's one way. Then, sorry, I actually wanted to talk about this one first. Then we can also mold the material. Think PLATO. Think, hey, maybe in that structure that I built all the way up, I pour some PLATO, and then it comes out with the shape that I'm interested in, like, you know, a cookie cutter of a sort, just 3D. Like you could be making a pyramid, for example, with different steps or whatever it is that you're interested in, you can build it that way. Or else we can remove material. We can use end mills, so like a drill, that's going to drill very, very, very small, and it's going to be automatized. So you can tell, okay, I want you to draw these 3 channels, and it's just going to do it for you. Differences. This one has a little bit less resolution. This one has a little bit more resolution, but it takes, well, this one you can do more stuff at one time, but it has like more steps. This one you only build one by one because you have to be drilling in every step of the way. So they're a little different. Some people might argue that 3D printing is missing from here. In my opinion, it's not there yet. It's coming up. So 3D printing is you take a material that can be molded and you sort of add like these new pens that you have that you can write in 3D, sort of that thing. The problem is we need a really high resolution to create these devices that I showed you. And the resolution of 3D printing is still a little on the higher end of things. So what it can produce is still a little large for what we need. So typically this is what we're going to be using more. And that's actually, that technique is called rapid prototyping. And it's why, one of the reasons why microfluidics has exploded. Because this is something that you can do in less than a day. And you try and create your own design from your brain to actually having a design in your hand. All right. So that technique that we talked about uses a material called polyphenol siloxane, which is a mouthful, so we call it PDMS. And I just want to spend a second talking about this. It has really good pros. So it's very transparent. Do you guys recall the glass plates that I showed around? The material that is inside is PDMS. And you can see through it, and it's very moldable. It has really good resolution. It lets oxygen go through, which is really important to keep things alive inside. But it's also hydrophobic. And that's a really important point, because as you very well know, cells have pieces that are hydrophilic, so soluble in water, and some others that are not. And some vitamins, for example, are soluble in water, some others are not. So maybe this guy at some point will be sequestering all of those that are not soluble in water. For you chemistry buffs, there's a lot of carbon, silicon, and oxygen over here. That's very hydrophobic. That's just a piece that changes to the other to make a polymer. So it's very hydrophobic, the result of it. Do we have any questions so far? All right, that's good. Okay, so here I have a very high resolution photograph of what the material looks like. Some of the edges aren't quite great, but for most of what we do, it's okay. You can see that this is 15 micron. A micron is a 1,000th of a millimeter. So that's tiny. So like sometimes, you know how you can see the topography differences in a coin. Some of the smaller ones might be on the upper end of the micrometer scale. Those could be like, I don't know, 100 to 200 micrometers, so that you can get an idea of what's the scale of those. It's this tiny. Okay. All right. Are we satisfied with the fabrication piece? Okay, cool. let's move on to the actual cancer that I told you guys that I was going to be talking about. So I'm gonna spend a minute talking about something called the tumor microenvironment. I don't know if you guys have heard about it before, but very briefly, it's the niche where a tumor develops. And there are a lot of cells happening over there. There are immune cells, there are fibroblasts, there are resident cells that just change things around. Some of those are going to be working for the team of the tumor. Some of those are going to be working in the other team. And sometimes it's really hard to know what is happening and how we can prevent that tumor from progressing, getting bigger, getting to the closest blood vessel, taking the tumor from there to another organ, and generating a metastasis, which is not great. So what we want to understand is how all of those players are working together or not, and how we can target them to make sure that that tumor doesn't grow. So something that I have been trying to convey piece by piece with all of this microfluidics talk is that we have devices that give us a lot of control over what we want to model. So there are a lot of pieces that are important in the tumor microenvironment. So it would be great if we could be using these devices and all of the advantages that they bring to create a complex environment and try to model what is going on over here. And that's precisely what we work on doing. We create 3D structures that resemble tumor microenvironments, that resemble blood vessels, that we can use to study what is going on. And then also other things that are very important are soluble factors. And some of those are like inflammatory factors, growth factors, and their concentrations are going to change over time and depending where you're at, which is what we call the gradient. And those are really important in cancer. So that's something that we really want to study. Signaling, so like how cell A talks to cell B by releasing a factor X, which could be an inflammatory factor. And that's something that we're going to be talking about. And something that I also want to argue is that, you know, your typical plate in vitro culture dish doesn't, like, it's not capable of doing any of this. And you'll see what I mean in a minute. Okay. So now I'm gonna be talking a little bit about the technology that you guys had in your hands. So this is what it looks like blown up quite a bit and in use. So what we aim to reproduce in most of these models is having a blood vessel or a lymphatic vessel, as I'm gonna talk about in a minute, and then cells around. And the way that we do it is by using 2 layers of PDMS, which now we all know what it is, that are sandwiching a rod of the same material. We inject in there a gel. So let's think about it like Jell-O, that is liquid first, and then it solidifies. And then we pull out that rod. To understand this, I typically tell people about taking a straw and putting it into a block of Jell-O and letting that Jell-O cool down. When that Jell-O is cooled down, you pull out that straw. You have a tubular structure in there. So think that we do that just a very, very small scale. We could put pieces of fruit into our Jell-O, something fun. So in this case, we could put different cell types in the hydrogel, in the scaffolding material, and then we could cover that tubular structure that we created with endothelial cells, which are those that compose the blood vessels or the lymphatic vessels. And there you have your first model. And the fun thing about this is that if you change the shape of the rod, you actually can produce different structures that look a lot like whatever your vessels in your body do. For example, this one is a lot of fun. I don't know if you guys can see it, but it actually branches down twice, like your trachea would branch out into the rest of the tree-like structure of your lungs. And that's something that, you know, it's 3D. Like it's actual tubes. You can't do that in a traditional petri dish. So there's where we get some of our advantages. All right. So first I'm going to be talking about the project that we have been working on for some time on breast cancer. I'm not the only one doing the work here. There's a lot of people, in particular, props to Dr. Vizamponik and now Dr. Karina Lucas-Intron, who was one of our students in the BB lab and has already graduated. So we're going to be talking about like how breast cancer, which is one of the most common cancer types in the US, typically generates in one of your mammary ducts, starts to grow, and then from there invades and gets out, tries to find your nearest vessel and tries to metastasize. And there are a lot of pieces that happen between point A and point B, between, hey, your cells are growing and proliferating in your mammary duct, and those cells getting out and finding the nearest vessel. And that's something that we're gonna talk about now. So the reason why I'm talking about metastasis is because, you know, it's said that women are going to have breast cancer in their lifetime. 90% of cancer-related deaths from those are going to be due to metastasis, not the actual cancer, but metastasis. So that's the reason why talking about metastasis and trying to understand metastasis, which right now we don't, is really important. Okay. So we have sort of modeled all the different steps that happen in the metastatic cascade in breast cancer, and yet we don't know so much about that. So here's the technology that I've been talking about to you guys. We have modeled pretty much every step that happens, and yet we don't really understand it. Something that we have been missing is talking about the lymphatic system, which you guys might have heard goes in parallel to your blood vessels, and is really not that well understood. Until recently, we had no real way to look at it quite well. So that's something that we have tried to do in the BB lab, and something that we have also tried to incorporate and look at that we know is important in breast cancer is collagen density. And I think you guys have heard about that before. So the reason why women get mammograms after a particular age is because an increased deposition of collagen in the breast is bad prognosis for breast cancer. So they typically, after that, you want to do additional screening. I'm not a physician, so that's for someone else to talk about. But what I can talk to you about is that increased collagen density is doing something to alter the game of the tumor. It's helping the tumor, it's playing the tumor's team. So we wanted to understand a little bit how. And to do that, we are going to be looking at lymphatic vessels. So the first thing we did is we created two different kinds of matrix. Let's think again about the jello, right? We're talking about making jello. We're making two kinds of jello, a concentrated jello and a less concentrated jello. And here is the difference between those. I don't know if you can see that, but this one looks a bit more porous and less robust than this one. So what we're going to be calling them is low density collagen, which I think you guys probably have heard about collagen by now, and high density collagen. So the low density collagen would be representative of a normal breast tissue, and the high density collagen would be representative of a tumor tissue in the breast. Something that, you know, if the tumor gets out, it's going to be bad. So there's what we're going to talk about right now. The first thing that we did is we used lymphatic cells to build a lymphatic vessel inside of our collagen gels. And as you can see, that definitely looks like a tubule. That's a very special kind of microscopy that does 3D renders. So I don't know if you can appreciate the 3D-like structure of that. It's kind of difficult from here. And then, so we know that those vessels express relevant markers that happen in vivo, those that we found about not very long ago that distinguish lymphatic vessels from regular blood vessels. Those are expressed when we make our vessels from scratch in the lab, which I think is really cool. But then what we found really interesting is that if we made these lymphatic vessels in the low density versus the high density collagen, after a while in the high density collagen, there started to be gaps. The vessels started to come undone, and we didn't know why. So we set out to investigate what was going on. The first thing that we did was we wondered, hey, is this really having an impact in how the lymphatic vessel would work? Does it matter, really? The first thing that a vessel does is it acts as a barrier. So a great way to test it is to add a fluorescent compound and track how it gets out little by little, like after a few minutes, after a few more extra minutes. So then what we saw when we looked at that is that in the high density collagen, that fluorescent compound got out of the vessel a lot quicker. So yes, there is a change that is happening in the barrier function in these lymphatic vessels in vitro. So then what we did is, okay, let's look at some factors that we know are can cause inflammation, and let's see if there's a difference between the high density vessels and the low density vessels. And we found that IL-6, those little asterisks mean that there's a really big difference. That was one of the ones that changed the most between the low and the high density vessels. So then we said, okay, is the density of collagen what is changing the secretion of these factors? So then what we did is we added a couple extra conditions inside. We had low density, which was 3 milligrams per ml, and then high density, which was 6. So we just added 2 in the middle to see whether it was really increasing. The concentration of IL-6 was increasing with the density of collagen. And as you can see, yes, not at first, but it does have an upward trend. So the more deposition of collagen there is, the more concentrated your collagen is, the more of this inflammatory cytokine that gets secreted by the vessel. So this is, by the way, the vessel talking to itself, which I think is really cool. So then we set out to try to understand whether this was like a cause and a causal relationship. So like, is the IL-6 responsible for this changing barrier function? So to do this, we did 2 experiments. The first one is we blocked that pathway of IL-6. So a little bit of background. You've got your IL-6, which is a soluble protein. Soluble proteins then bind to a receptor, and then afterward, things happen. Like other proteins get activated, and then things happen in the system. It really depends on the pathway. And we really don't know if that is what is going on. In this case, what we're hypothesizing is IL-6 binds to a receptor, and after a while, the barrier function changes, right? So what we did is we said, okay, let's use an antibody to block the IL-6 receptor and see whether, if we block that, the high density condition will level down to the low density condition. And that's actually what happened. Yes?
Where are the IL-6 receptors located?
So those are typically on the outside of the cell.
They're on the outside of the blood vessel cells, or
yes,
other kinds of cells?
Other kinds of cells as well.
Okay.
But in this case, we're only talking about the lymphatic cells because there are no other cells present in our system. So there's nothing else that could be messing up with that. The only thing that we're changing is the concentration of the collagen, which I think is bananas, by the way. I really like my work. All right, so then we did the opposite thing. We said, okay, so if we add IL-6 to the low density condition, is it going to rise up to the high density condition? Is that going to be the sole factor or one of the contributing factors, the major factors that is going to level that up? And the answer is yes. IL-6 is doing most of the work in changing the barrier function of the lymphatic vessels. So there's that, one mystery solved. Now you might say, hey, Maria, you said that we were going to talk about cancer. Yes. So then what happens if we put our breast cancer cells around that lymphatic vessel? And what we saw was over here that when you add the breast cancer cells, which is that extra condition over here, the barrier function was worse only in the low density condition. Only in the low density condition. So there's that. We said, okay, so what we can do is we can check whether IL-6 is still responsible for this. And maybe that's what is changing between these 2 conditions. So we did that again. We said, okay, let's add our antibody that is blocking the IL-6 receptor. And over here we saw that, yes, it goes down, not all the way down to the low density condition. So there's other things that are playing a role over there. And this story actually ends over here, because this was one of the first studies that we did using the system. We're hoping to do more on this and to figure out what else is drawing that. We already have a couple of candidates and we're looking into those, but we still don't know what it is in that crosstalk that is changing the permeability of those vessels. So that's story number one. All right, we're going to switch gears a little bit. We're still going to talk about lymphatic vessels, so let's not forget about those. But now we're going to be talking about head and neck cancer. Again, props to Karina, who did most of the hard work in here, and to Dr. Paul Ferrari, who was a great source of knowledge for this project. All right, so head and neck cancer. This is an umbrella term to talk about every tumor that happens in the nasal cavity, the mouth cavity, the pharynx, everything in there. So it's no surprise that you see that patients have like really diverse tumors that, you know, patient A, their pathology looks nothing like patient B's. They're really heterogeneous in themselves. But they do overall amount to like 4% of cancers in the US, which is a lot. And yeah, they're very complex, they're very heterogeneous. What we do know, and we have seen in the clinic and in animal models, is that the lymphatic vessels, which by the way are all around your head, and there's a lot of lymphatic nodes as well, are very possibly involved because we tend to find cancer cells in there. And we also know that the prognosis of these patients tends to be worse when there's lymphangiogenesis. So angiogenesis of the lymphatic vessels, which means that the lymphatic vessels grow and make new branches and new vessels overall. Those tend to grow and the patients are going to do worse. And we don't really know what. We have some ideas, and one of them are cancer-associated fibroblasts. These are cells, they're just around, they're the doers in the tissues. They change things around, they signal around. We don't know how, but we think they're contributing to generating these angiogenesis of the lymphatic vessels. But we don't really know how that works. And if that's all for the same patients, all the same for them, or if that is a source of variability that is happening among the different patients. So that's something that we wanted to investigate using pretty much the same model that you guys saw a minute ago. So all the same concepts are going to apply. So since we work by the hospital, we had access to residual tissue of resections of tumors from head and neck patients. So we have 3 patients of head and neck that we knew that they had fibroblasts in their tissue because those purple things over here, over there, and over there, those are consistent with the presence of fibroblasts in the tissue. So we knew that very likely there were fibroblasts in there. So the next thing that we did is we took another piece of tissue that we had, and then we extracted fibroblasts. So we mix it up in the lab with different things, and then we try to culture them. And then after a while, we come up with a set of fibroblasts, a lot of them, that we can use to study. So then what we did is, instead of using the breast cancer cells as before, we substituted those by those cancer-associated fibroblasts from the different patients. So you have your vessel and your fibroblasts from patient 1, patient 2, or patient 3. The lymphatic vessel is the same for all of them. So we're going to see how they talk to one another. I think that's a very important point. The lymphatic vessel is always the same. So what we did is, again, we looked at the barrier function, but we also looked at the branches. Because after 5 days, if you culture those 2 together, am I over?
No, you can go
It's okay. Yep, that's all right. Don't worry. It's 2 o'clock, but it's
After 5 days, there's a lot of branches that appear and that look a lot like a lymphangiogenic sprout that I like to call. So we started to investigate those angiogenic sprouts for the different patients. And we saw that. I'm just showing you guys the lymphatic vessel in this case. So you can see that the branches are a lot bigger in the sample for this patient, in the lymphatic vessels for patient 1 than for patient 3. So we actually quantified that, and we looked at how many there were and how long there were. And as you can see, there's already some variability happening. So they're talking to the lymphatic vessel in different ways, depending on which fibroblast you're talking about. So you can see, for example, patient 1 had the longer and the most abundant sprouts, whereas patient 3 had less of them and they were shorter. But still, always they were higher than in the lymphatic vessel alone with no fibroblast, which I think is really cool. All right, so the next thing we did is we looked at the barrier function, because we've already established that's something that we like a lot. And it's really easy to do in this system. So the first thing that I want to show you is that when we have no lymphatic vessel, just the collagen, yellow, as we've been calling it, the barrier function is like nothing. This is your baseline. There is nothing preventing that fluorescent molecule from flowing outside. However, if we have our lymphatic vessel, it retains that fluorescent molecule a lot. Now, if we do those co-cultures of having the lymphatic vessel plus the fibroblasts, we see that for 2 of the patients, the barrier function was worse. Interestingly, not for the other one. So we were very intrigued and we wanted to have a look at what was going on. So what we did is we separated the lymphatic vessels from the fibroblasts, and we did an RNA screening. When we talk about RNA screening, it's a hard concept. It's sort of looking at what the cell is thinking about doing, but not really about what it's doing. You know, there's a lot of things that are happening, lots of thoughts. So that's what we're gonna look at. I'm sorry, there's a lot of information, but I couldn't get this organized better. I couldn't animate it. I couldn't. All right. So here is a representation of the things that were changed between the lymphatic vessel alone and when it was in the presence of those fibroblasts. And each of these colors represents what was going on in one of the patients. What were those changes in each of the patients? The names that are in the center are the genes that were dysregulated, so like messed up in all of them. And the other ones were sort of like shared between 2 of them, but not the other. Over here, 2 of them, but not the other, which I thought was very interesting. So some examples for like, you know, for mentioning something are this guy over here, which is integrin beta 3, which we know is related to the metastatic process. And it's basically a protein that helps cells anchor to things. So you would think, okay, if there's branches, it's probably because the cells are anchoring more to things. So that one was up for patient 1. Remember, that was a patient that had most branches and the longest ones. So that makes sense to us. Another one over here is something called a Metalloproteinase Inhibitor. Metalloproteinase, it basically means that the cell is releasing chemicals to break down the jello and get in there. So this molecule actually inhibits it, but it's downregulated. So the cell is secreting something. All right, let's see if I can say this right. The cell is not secreting something that will prevent it from breaking down the jello. That's a fault of the cell. Something else would be MMP9, which is one of those metalloproteinases. So those that do get broken down, those that are breaking down the collagen. So those are also differentially expressed, which I thought was interesting as well. And then CDH1, that's one of my favorites. That means that the cells in the vessel are talking less to one another. They, instead of like creating a monolayer, so think of a rug, that rug has little holes. So that means that when we add that fluorescent compound, if that molecule is down, that means that it can probably more easily get out of the vessel and have a lower barrier function. Is that making sense? So we do see some things that may be changing and maybe explaining what are some of the changes that we see differentially in the different patients. Now, something that I thought was really interesting is that the expression profile and the functional responses were consistent with the tumor grade. So we saw that, for example, the tumor grade was higher for patient 1. Remember, that's the patient that actually had the longer and most abundant sprouts. So we're already seeing things that are consistent with what the doctor saw in the patient, indicating that this could be a technology that could be really interesting to figure out how the patient is going to be doing. And the thing that I'm going to talk about next is, could we use this technology to actually pick what is some therapy that would be useful for them? So something that we looked at, this IGF-1 frame over there, is the gene that was dysregulated in all of the patients. So we said, okay, there are inhibitors for that gene, and we know it's a growth factor. So if there's less growth because we cut down that growth factor, maybe we can stop that lymph angiogenesis from happening. So we did that. And we treated our models with the fibroblasts and the lymphatic vessels with an anti-IGF-1 treatment. And interestingly, it didn't work for some of our patients. It did work for others. We don't really know exactly why, because probably the thoughts of the cell are way too hard for us to comprehend. But we do have an idea of, hey, are they really going to respond? Yes or no? For example, over here, the permeability. You can see over here that for patient 1, the permeability did go down. So that means that it was restored. They had a more less permeable lymphatic vessel, but they did have more sprouts than before, which is not great. We don't really know how. We do know that this technology is really powerful, and we definitely want to keep looking this way. All right, take-home messages. So we have been talking about microfluidics and how we can use it to integrate more things into our models to try to get a more accurate response and to try to understand better how those certain cells are playing for the tumor side, certain cells are playing against the tumor, to try to understand that better. And perhaps that way we can understand how to treat it better. Not intended to replace other methods of studying things, but to complement. And well, we have been talking especially about lymphatic cells. There is very little research in this field. We're actually one of the few labs that is looking to understand how lymphatic vessels work in a more generalistic way. So we're hoping that we'll learn how to better target lymphatic cells in the future. And yeah, try to put a little grain of salt into research for cancer. And that's everything that I have for you guys. Acknowledgements. Everyone else in my lab, funding agencies, everyone who's giving us a hand in figuring out how this works. And you guys for bearing with me for a whole hour. Thank you so much, and I will be happy to take your questions.
I remember when I was back in school, macrofluidics was it. And you know, they had differential equations they tried to do, impossible to solve, but they kind of simplified it. And now they can do the powerful computers at it, so like weather forecasting, boats in water. It's pretty darn good results. Is there any kind of modeling that can go on with microfluidics to predict how things will happen?
It all comes down to what it is that you want to look at. For example, one of our people in the lab has been very interested in looking at how oxygen is in some parts of the tumor and not really in others. And that's something that you can model better than measure. So that's something that has been looked at. And there are really very interesting results in that case. Something else that really gets modeled more than measured is rate of flow. We haven't really talked about that, but you could actually flow liquid through our vessels like in reality, right? So you could measure how that changes fluid dynamics in our system. you could measure some things, some others you would model, and you could model how if you change the rate of flow a little bit, what are the things that will change in your system? So yeah, there's really cool work out there. It's just not necessarily my expertise. There's that. Do we have any more questions?
I have a question from the chat on Zoom. Sandy Rotter was saying, I wonder if you work with the UW-Madison Departments of Biomedical Engineering, and they mentioned a talk specifically on engineering microenvironments to regulate forces, form, and multicellular function. So I think just wondering in general if you work with the, yeah. Oh yeah, you can see the chat too, that's true.
Oh yeah, here you go. Interesting. So Dr. Christopher Chen is actually one of the people who does work very similar to this. He works a lot on vascular biology and how that can be targeted for specific diseases or how that changes depending on diseases. He has really interesting work, very detailed. It goes down to the molecular biology a lot. I really admire his work. I think he's great. I did not know that there was a talk by him. Plus he's a delightful human. So if you guys get a chance to log in there, I would encourage it. I think it's probably going to be very exciting. Yes. Hi.
Not coming from a science background.
That's okay.
How does the lymph system work? Does it go through the blood? Does it?
It does not. It sort of runs parallel to the blood. What it does is
it have its own vessels?
It does have vessels. So it kind of works the other way. So you think about the blood vessels running and delivering nutrients and blood. Mm-hmm. What they do is they pick up the trash. The lymphatic vessels pick up the trash. And they gather everything and they take it to the lymphatic nodes. Mm-hmm. And in that trash, sometimes what there is are pieces of like bacteria and such, so that our immune cells can recognize those. Mm-hmm. And it's sort of like those wanted posters. It's exactly the same.
And people with more sprouts, is that a bad?
It has been shown to be correlated with worse prognosis. So that means that typically people who have lymph-angiogenesis in their tissue, they're probably going to be doing worse. And
those most sprouts, do they have anything about dissemination of the cancer cells through the?
That is a great question. I think probably so, but we don't know enough. The problem is we always look at the final picture. You see, we look at, hey, there's more, but we don't know how we got there. Mm-hmm. And that's something that we're trying to look at these systems. Like, okay, what happens?
So the sprouts, do they break off and go?
The idea is that they don't, they just generate a new vessel piece. Mm-hmm. So what we know, for example, about blood vessels is that they create new pieces of vessel and they get sequestered by the tumor to get more food. So perhaps there's something to that, similar for the lymphatic system. We just don't know enough because we haven't really had the tools to look at them until very recently. Yes.
Yeah, you mentioned the variability in the way that different patients will respond to different treatments and can't really predict it in kind of an analytical way, but is there an attempt to maybe like create like a massive database where you can apply some AI tools to suggest what the best treatment might be for a given patient?
There is. I actually just wrote a small paper on this, so I'm kind of up to date with this. It really depends on the tumor. For some of those, if you look at what kind of tumor the patient has, you're able to say, okay, they're going to respond really well to this. For some others, no, it just doesn't work. There is nothing up to now, no biomarkers, which is what we like to call, that are going to tell you, hey, this person is going to respond to this. And that's the challenge. Mm-hmm. But yeah, we're getting there. Perhaps AI will help us with some of that. We're not really sure as of now. this is one way that we're collecting more data to try to understand these systems better, to see if we can understand how that variability could be coming from different moves that the tumor could be doing, if that makes sense.
How long in the future do you see yourself in this area?
I think quite a bit. I'm hoping so. I think there's still a lot of work to be done. So very many different tumors, so many complex interactions that we could look at. So we haven't really spoken about that, but the role of the immune system in many of this situations is really important. Yet we haven't really integrated it very well into our models just yet. And I think that's one part of the field that is gonna start developing more in upcoming years.
Well, you'll have to come back and tell us about it.
Will do, thank you. That makes me happy that you guys like this.
Thank you. Well, thank you for that.
Very much.